Viruses in molecular biology. In this chapter, we explore the world of viruses, focusing on bacteriophages and animal viruses. By the end, you'll be able to compare and contrast the shape, life cycle, and replication mechanisms of various bacteriophages. Describe the general features of animal viruses, predict their mutation rates based on genetic material, and analyze the genome organization and replication strategies of retroviruses. Bacteriophages, or phages, were first studied for their potential as antibacterial therapies. They were discovered as filterable agents that killed bacteria, particularly in soldiers with dysentery. In the 1940s, researchers like Max Delbruck and Salvador Luria led a group studying phages isolated from raw sewage. The most studied were the T phages, including T2, T4, and T6, collectively known as the T-even phages. This figure shows the relative sizes of viruses compared to a eukaryotic cell, its nucleus, and the bacterium E. coli. Viruses are microscopic entities, smaller than cells, and their scale helps us appreciate the simplicity and compact nature of viral genomes. Bacteriophages are categorized into three morphological classes. First, we have icosahedral tailless phages. Second, icosahedral phages with tails, like the T-even group. And third, filamentous phages. Each structure influences how the phage attaches to and penetrates the host cell. Phages can follow one of three life cycles. In the lytic cycle, the virus hijacks the host machinery, replicates, and causes lysis, releasing hundreds of new phage particles. The lysogenic cycle integrates phage DNA into the bacterial chromosome as a prophage, allowing dormant replication. Chronic infections allow phages to continuously bud from the host cell without killing it. Bacteriophages form plaques, clear zones, on a lawn of bacterial growth. Each plaque originates from a single phage particle. This method allows for precise quantification of viral particles and isolation of specific phage strains. Even phages like T2 and T4 are virulent bacteriophages. They inject linear double-stranded DNA into the host, which contains a unique modified base, 5-HMC, instead of guanine. Their DNA is terminally redundant and circularly permuted, features that assist in efficient packaging and protection from host enzymes. The lytic cycle of T-even phages begins with adsorption and DNA injection. This halts host gene expression, allowing phage genes to take over. Enzymes are produced, DNA is replicated, and new virus particles are assembled. Finally, the host cell bursts open, releasing the new virions. Let's look more closely at the T-even life cycle. Phage DNA replicates as concatenars, long, repeated DNA units. Genes for head and tail structures are transcribed and translated. Terminase cuts and packages DNA into pre-assembled heads. Endolysin and wholin work together to degrade the cell wall, triggering lysis. This slide outlines the timing of each stage in the T-even lytic cycle, from initial adsorption and enzyme production to DNA replication, particle assembly, and cell lysis. Understanding the timing helps us map the expression pattern of phage genes. Phage T7 has an icosahedral head and a short tail. Unlike T-even phages, its DNA is not circularly permuted. It enters the host using both host and its own RNA polymerases. As it replicates, the DNA forms concatenars, which are cut into genome-length units for packaging. This figure breaks down T7's replication timing. Early gene expression is driven by host polymerase, but later, the phage's own polymerase takes over. This tightly controlled switch ensures efficient replication and viral particle production. X174 is a virulent phage with single-stranded circular DNA. The strand within the virus is the positive strand. Host enzymes convert this into a double-stranded replicative form. The negative strand is transcribed and translated to support viral replication. RNA phages like MS2 and Q carry single-stranded RNA as their genome. The RNA acts directly as messenger RNA. These phages use an RNA-dependent RNA polymerase to replicate their genome by first synthesizing a complementary negative strand, which then serves as a template for producing new positive strands. Temperate phages, like lambda phage, do not immediately destroy their host. Instead, they integrate their DNA into the host genome and replicate silently as prophages. In the lambda phage, genes are arranged in functional clusters. This integration allows the phage to persist within the host and switch to the lytic cycle when conditions favor it. After lambda phage DNA enters the host cell, it forms a circular structure by base pairing between its cohesive ends, called cos elements. This circular DNA serves as the foundation for either entering the lytic cycle or integrating into the host genome as a prophage. This diagram illustrates the dual path lambda phage can take. In the lytic cycle, viral replication begins immediately, ultimately leading to host cell lysis. In contrast, the lysogenic cycle involves the integration of lambda DNA into a specific site in the host chromosome. The integrated DNA remains stable and can switch to the lytic pathway under certain conditions. In the lytic pathway, lambda DNA first replicates using theta replication to increase copy number, followed by rolling circle replication that generates concatenators for packaging. During lysogeny, the DNA integrates into the host genome at a specific site, forming a stable lambda lysogen. This lysogen can re-enter the lytic cycle if triggered by stress or environmental cues. P1 is another temperate phage that can undergo both lytic and lysogenic cycles. Structurally, P1 has an icosahedral head and a complex tail with a contractile sheath. Its linear double-stranded DNA is terminally redundant and circularly permuted. Notably, P1 can infect a broad range of gram-negative bacteria. In the lytic phase, P1 DNA replicates using theta and rolling circle mechanisms. The DNA is cut at a pack site, packaged into capsids, and then cleaved again when the head is full. In lysogeny, P1 DNA exists as a plasmid, maintained at 1 to 2 copies per cell. This plasmid form allows P1 to mediate transduction, transferring bacterial genes between hosts. Chronic phages such as M13, F, and F1 establish long-term infections without lysing the host. They program the host to continuously release new virions. Upon injection, their single-stranded DNA forms a stable hairpin, serving as a primer for synthesizing the complementary strand. These phages bud off from the host cell surface indefinitely. Now we turn to animal viruses, focusing on 3 key types: Polyomaviruses, adenoviruses, and retroviruses. Animal viruses vary by genome type, either DNA or RNA, single or double-stranded, and by whether they are unclosed solely by a protein capsid or an additional lipid envelope. Polyomaviruses contain small, circular double-stranded DNA genomes and have icosahedral capsids. SV40, a polyomavirus found in monkeys, has a genome divided into regulatory, early, and late regions. Its capsid contains 3 structural proteins, VP1, VP2, and VP3. In SV40, the early region encodes large and small T antigens. The large T antigen is essential for initiating DNA replication and disrupts tumor suppressor genes. The late region contains genes for capsid proteins. DNA synthesis is limited to the S phase of the host cell cycle. Adenoviruses are linear, double-stranded DNA viruses with blunt ends and inverted terminal repeats. Their large icosahedral capsids contain genomes of about 35 to 45 kilobases. Because of their ability to deliver foreign DNA into host cells, adenoviruses are widely used as vectors in gene therapy, although pre-existing immunity can limit effectiveness. Retroviruses are RNA viruses that replicate through a DNA intermediate. They use reverse transcriptase to convert their RNA genome into DNA, which then integrates into the host genome. All retroviruses carry two identical RNA strands and three essential coding regions: GAG, POL, and ENV. HIV, the virus responsible for AIDS, is a notable retrovirus. The GAG gene produces polyproteins cleaved into matrix, capsid, and nucleoprotein components. POL codes for reverse transcriptase and integrase, enabling the viral DNA to integrate into the host genome. ENV codes for transmembrane proteins critical for viral entry. Protease activity is essential for cleaving these polyproteins into their functional forms, and protease inhibitors are key drugs in AIDS therapy. Retroviral replication includes early and late stages. In the early stage, reverse transcriptase creates a complementary DNA strand from RNA, then degrades the RNA and synthesizes the second DNA strand. This double-stranded DNA enters the nucleus and integrates into the host genome via integrase. In the late stage, the provirus is transcribed and translated to produce new virions. A few important notes. HIV is a virus, human immunodeficiency virus, while AIDS is the resulting disease. RNA viruses mutate rapidly, complicating treatment and vaccine development. Because AIDS lacks an animal model, testing therapies is particularly challenging. To summarize, retroviruses are a unique class of RNA viruses that integrate into host genomes. Their replication, high mutation rate, and capacity to persist in a latent state make them central subjects of biomedical research and therapeutic innovation.